Backcross. In backcross, F1 hybrid is crossed back with any of the parent, either dominant or recessive. Backcrosses are used to increase useful traits in a population. So before learning about backcross, we need to know that who was the one who proposed backcross. Backcross was firstly proposed by the scientist named Borlaug. This backcross is basically done to increase the useful traits or a useful character in a population of any plant or any animal. So in this method, the F1 hybrid is backcrossed with any of the parent, either with the homozygous recessive or either with the homozygous dominant. For example, certain crop plant hybrids are backcrossed with wild species to obtain its useful traits such as disease resistance. Suppose parental generation के किसी plant में एक trait था disease resistance and we want many more species to have this trait. क्योंकि अगर कोई प्लांट डिजीज़ रेजिस्टेंट होगा तो उसे कोई भी पैथोजन उतना ईज़िली अटैक नहीं कर पाएगा और इससे जो क्रॉप है वो काफ़ी अच्छी क्वालिटी का होगा. सो फॉर हैविंग मैनी मोर नंबर ऑफ स्पीशीज़ टू हैव दिस ट्रेट वी विल सिंपली बैक क्रॉस दी plant of F1 generation with the parental generation, with either of the parental generation which will have this trait. Let us now come to an example. So here we can see that in a parental generation, the tall plant and the dwarf plant was crossed, and in F1 generation we got heterozygous dominant plant. So this heterozygous dominant plant will be backcrossed with either of the parent. But here I have backcrossed the heterozygous dominant F1 generation plant with the homozygous dominant, and after the backcrossing we get capital T which is a tall plant, and capital T which is also a tall plant, and two heterozygous dominant, which means that they are present in a equal ratio after backcrossing. We can also backcross the heterozygous dominant plant with homozygous recessive of parental generation plant, but we will do it afterwards, because if we will cross the F1 generation plant with the homozygous recessive of parental generation, it will known as a test cross. It will also be a back cross, but it will be named as a test cross because this method is using to test something, whereas the back cross was done to produce the desirable character in an upcoming generation. A test cross involves meeting of an unknown genotypic individual with a known homozygous recessive. This is because recessive alleles will always be masked by the presence of dominant alleles. Hence, the phenotype of an offspring will reflect the genotype of the unknown parent. Basically, this test cross is done to test the genotype of a particular plant. Suppose the plant is in a dwarf condition, then we know that this plant is in a homozygous condition because a dwarf plant is of a recessive character, expresses a recessive character, and it can't be heterozygous because heterozygous plants are always dominant one. So if we need to test the genotype of a dominant plant, so we need to do a test cross because we don't know that if this particular dominant plant is having a homozygous condition or having a heterozygous condition, because it can be either homozygous or it can be either heterozygous. Here we can see that the heterozygous plant of F1 generation is crossed with the homozygous recessive of a parental plant. So this is also a back cross because the offspring generation plant is crossing with the parental generation plant. Now let's suppose we have to test the genotype of this particular plant. Here the genotype is written, but we have to suppose that we don't know the genotype of this particular plant. So we will cross it with the homozygous recessive of a parental plant, and if after crossing the plants will be in an equal ratio of both the character, then we can say that the plant which was taken to test the genotype, that plant was the heterozygous dominant. Now let's see the another case. In this case, the taken plant was also tall, because we want to test its genotype, but we know the phenotype of the particular plant, that it is tall. We will cross it with the homozygous recessive of a parental generation, and if the offspring will all be tall, then it can be said that the taken plant was a homozygous dominant, because whenever we cross one pure homozygous dominant with the pure homozygous recessive, the all kind of offspring we get is only a heterozygous dominant. And here, if the offsprings are all tall, then we can conclude that the taken plant was homozygous dominant. But if the offspring will contain both the character of parental generation in equal ratio, then we can say that the taken plant was heterozygous dominant.